Good morning, everyone, and thank you for joining us for Winship Grand Rounds. If you're an Emory University or healthcare employee and want to get CME credit for attending today, if you click on the chat tab at the bottom of the screen, Julie will give you a link to log in and get CME credits. And if you have any issues with the CME login, email Julie or send her a note by the chat and she'll help you out. This morning, it's really a special pleasure to welcome Yvonne Maillard. Yvonne is a professor at the Perelman School of Medicine at the University of Pennsylvania. He went to medical school in the University of Lausanne and Zurich and got his MD, PhD degree at the Swiss Academy of Medical Sciences. He completed his residency in Lausanne and then did his fellowship at UPenn, where he's built a translational research career and has risen to be full professor and vice chair for research. His laboratory investigates the regulation of normal and malignant hematopoiesis, bone marrow transplant, and T-cell alloimmunity, and he's focused on the role of Notch signaling, really I think one of the most interesting proteins and signaling pathways that has been discovered in immunology. And how Notch signaling regulates T-cell development, differentiation, and functioning. He's developed really some very exciting and translatable insights into how Notch receptors and their ligands regulate graft-versus-host disease in mouse models and in human disease. And I think this is a great example of translational research at its best, from basic science discoveries into clinical translation. He's also looked at the role of the trithorax family and epigenetic modifications of the genome and how that affects hematopoiesis and leukemia. Welcome, Dr. Maillard.
Well, thank you very much, Ned, for this kind invitation and for all of you to share some time this morning with me. It's a pleasure to be here virtually, and I'd like to share some of the work in my lab studying the role of Notch signaling and specifically Notch ligands expressed by a new population of stromal cells in secondary lymphoid organs, both in the context of graft-versus-host disease, which has been a key interest in my lab for a long time, but also in other contexts of lymphoid homeostasis and lymphoma pathogenesis. So basically new roles for these unique types of stromal cells expressing Notch ligands in hematology. This is the menu of my presentation today. I will start by summarizing the body of work that we have performed so far and ongoing work in my lab about the role of Notch signaling in T cells to drive graft-versus-host disease pathogenesis after bone marrow transplantation. This is both in mice, and now we have data in non-human primates as well that I will share with you. I will focus the next part of the talk on describing this discovery that we made. It really was surprising to us at the beginning that the critical source of Notch ligands in graft-versus-host disease appears to be a non-hematopoietic fibroblastic stromal cell type in secondary lymphoid organs very early after bone marrow transplantation. And then in a second part of the talk, I will describe new emerging data from my lab and from other labs suggesting that these stromal ligands are also very important to sustain normal and malignant B cells. I'll show you some data about their role in sustaining mature B cell subsets in steady state and in lymphopenic conditions. And I will allude to data about B cell lymphoproliferative disorders such as chronic lymphocytic leukemia and marginal zone lymphoma, which are commonly driven by Notch signaling. So in essence, my lab studies a pathway that was first identified in the fruit fly about 100 years ago. We model the effects of this pathway in mice. We use non-human primates selectively to do advanced preclinical translational work, and we have our sights on human applications of our basic science discoveries. So in terms of graft-versus-host disease, I want to briefly remind this audience that acute and chronic graft-versus-host disease after allogeneic hematopoietic cell transplantation remain very common problems in 2021 for our patients undergoing bone marrow transplantation or hematopoietic cell transplantation. It is a donor T cell mediated disease at least at early time points that targets multiple organs, including the skin, the intestine, the liver, and the lung. And both the acute and the chronic forms of the disease remain major clinical problems. Some of the major unmet needs are summarized on this slide for those of you who don't see patients with graft-versus-host disease every day. One of the main problems is that we prophylax all the patients with global immunosuppressive drugs, yet this prophylaxis is often ineffective and patients still have a high incidence of acute and chronic graft-versus-host disease. There's a very high mortality in patients who have severe grade 3, grade 4 graft-versus-host disease who fail corticosteroid therapy. And in these patients, intestinal graft-versus-host disease is most often the central life-threatening complication. I'm making that point because you will see that one of the key activities of Notch blockade is to actually prevent intestinal graft-versus-host disease. And then last but not least, there is a huge morbidity of chronic graft-versus-host disease in a high fraction of our patients, even when they have been cured of their underlying hematological disorder or blood cancer, which can alter their life essentially for a very long time. So there is a need for both better prevention and better treatment of graft-versus-host disease, all of this without eliminating the key anti-tumor effects of bone marrow transplantation, which is the reason why the procedure is being performed in the majority of cases. So we think that Notch is a major regulator of graft-versus-host disease. I want to introduce you to this pathway, which I think, like Ned, is a really interesting pathway in immunology and hematology. So it is a ligand receptor pathway that operates between adjacent cells. Mammals have a series of 5 different Notch ligands that are like 1, 3, 4, jagged 1 and jagged 2 that signal with 4 different Notch receptors. Ligand receptor binding triggers a series of proteolytic cleavage steps that ultimately release the intracellular domain of Notch into the cytoplasm. Intracellular Notch can then migrate into the nucleus, where it becomes part of a large transcriptional activation complex in association with a transcription factor partner called CSL, or also called RBBJ kappa. And that recruits a co-activator called mastermind and an additional series of co-activators that eventually turn on the activity of transcriptional targets in a very context-dependent fashion. And this is a pathway that's very conserved during evolution that links an extracellular signal to transcriptional activity in the absence of complex signal transduction steps. Simply the cleavage of the intracellular domain of Notch gives rise to something that will become essentially a transcription factor. So Notch plays a lot of functions in many different contexts, but my lab has focused now for 15 years or so on the role of Notch signaling in mature T cells. And to address that function of Notch, we have been agnostic initially to the nature and the source of Notch ligands in vivo by restricting our work to in vivo studies using loss of function, genetic and pharmacological approaches, and looking at very specific immune contexts. And taking that approach, we have uncovered major functions for the Notch signaling pathway in a range of immunological conditions that have high relevance to human disease. And they include acute graft-versus-host disease, which I will talk about today, but also chronic graft-versus-host disease in work that we have performed together with Stephanie Sarantopoulos at Duke and Bruce Blazer at the University of Minnesota. We've also done work that I'm not going to talk about today in experimental immune encephalomyelitis, so a model of multiple sclerosis, and in heart transplant rejection, indicating that the roles of Notch signaling are not limited to graft-versus-host disease, but yet GVH will be the focus of my talk today. So our initial working model when we started this work is that Notch ligands will be provided by host antigen-presenting cells and potentially upregulated by the effects of inflammation and conditioning. and that they would interact with Notch receptors in incoming naive alloreactive T cells around the same time as they interact with antigen-presenting cells presenting alloantigen and co-stimulatory molecules. And we thought that this may have a major impact on the effective T cell differentiation out of these naive T cells. So you'll see later on that we have now revised this model to include this additional cell type, which we think is critical, this fibroblastic stromal cell type, which we think is critical. And I'll show you how we came to the conclusion that the model needed to be revised in that fashion. But we have used a series of mouse models to study the role of Notch signaling in graft-versus-host disease. And one of them that is central to our studies is summarized on that slide. In this case, I'm showing you an MHC mismatch model of bone marrow transplantation. It would roughly approximate a haploidentical model of bone marrow transplantation in humans without immune prophylaxis of graft-versus-host disease. We use biopsy mice as recipient after lethal irradiation. We reconstitute them with T-cell depleted allogeneic bone marrow from C57 black 6 mice. And then we add or do not add well-defined subsets and numbers of wild-type T cells or T cells that are genetically engineered to lack the ability to respond to the Notch signaling pathway. And as a comparison, we can compare the effects of this genetic intervention with pharmacological interventions that block individual Notch receptors or individual Notch ligands with monoclonal antibodies. And this has been a wonderful partnership with Chris Siebel at Genentech, using antibodies that cross-react against mouse and human Notch receptors and ligands. So it can be studied in mice, but potentially considered for human applications. We can assess the effects of these interventions on survival, on the severity of graft-versus-host disease, and on T cell expansion, differentiation, and effector functions. So this is the results of one of our initial experiments that is published, but that I want to highlight here because it really triggered our deep interest in the activity of Notch signaling in this context. So this is simply showing survival of 3 cohorts of mice after this MHC mismatched transplantation in the B62 biopsy model. Over time after transplantation, you can see that when you don't transplant T cells, you just do a transplant with T cell depleted marrow, the mice survive, they reconstitute their hematopoietic system, they don't get graft-versus-host disease. When you add wild-type T cells into the mix, you get this very high and profound lethality that occurs within the first 30 to 40 days after transplantation in this very severe model of graft-versus-host disease, as reported by many others. But strikingly, when you transplant these mice with T cells that have been genetically engineered to fail to respond to Notch signaling, we can rescue this high mortality back to the low mortality that you see when you do a transplant without any addition of T cells. So a major shift in survival that suggests a major activity of the Notch pathway in graft-versus-host disease. The T cells that we are using here are building on a genetic trick where we express a dominant negative inhibitor version of this co-activator mastermind I mentioned in one of the introductory slides, which locks the transcriptional activation complex in the off state downstream of all receptors and ligands. And together with that, we have documented markedly decreased graft-versus-host severity, graft-versus-host disease severity using pathological criteria, markedly decreased production of many inflammatory cytokines, but interestingly preserved in vivo T cell expansion in lymphoid organs and actually enhanced expansion of Tregs both in lymphoid organs and in target organs. We have also, in data that I'm not showing you for the sake of time, and I'm mentioning that for some of the transplantation aficionados that might be in the audience, we've validated these results in a range of other models of graft-versus-host disease in mice, parent to infant models, with and without irradiation, minor alloantigen mismatch model, chronic graft-versus-host disease model. We've seen major effects of Notch in all of these contexts really suggesting that it is a central pathway in regulating the pathogenesis of graft-versus-host disease in mice. Now, what was interesting about the activity of Notch blockade in T cells is that we observed highly preserved graft-versus-tumor activity after transplantation of Notch-deprived T cells, dominant negative mastermind expressing T cells, or T cells that were engineered in other ways to fail to respond to Notch signaling. This is shown here on the left with a study where we inject the recipient mice with luciferase label A20 leukemia lymphoma cells that mimic the situation of patient with residual leukemia at the time of transplant. You can see in the absence of T cells, rapid leukemia progression as indicated by bioluminescence imaging. In the presence of wild-type T cells, we control the leukemia with no residual luciferase signal at day 14, but the mice die rapidly thereafter of severe graft-versus-host disease, as I've shown you before. But interestingly, when we transplant dominant negative mastermind expressing T cells that cannot respond to Notch signaling, we preserve the graft-versus leukemia activity to a large extent, end up eliminating all the luciferase activity in this particular model, and we don't induce severe graft-versus-host disease. So we end up observing long-term leukemia-free and graft-versus-host disease-free survival in mice, a very desirable outcome that we would want to achieve, of course, in our patients. And what we can see also is that when we test the cytotoxic activity of our dominant negative mastermind expressing T cells, in this case, in this ex vivo assay, but we have several in vivo assays as well, we can see that the dominant negative mastermind expressing T cells perform as well as the wild-type T cells, or nearly as well as the wild-type T cells, in this case on the left against two model leukemia line. There's a negative control on the right. And so their graft-versus leukemia activity is much better preserved than their capacity to induce graft-versus-host disease. In other words, immunomodulation, but not global immunosuppression. In this slide, for the sake of time, again, I'm summarizing a whole range of published findings from the lab that have delved into the mechanisms of this effect. We see that Notch blockade blunts the pathogenicity of alloreactive CD4 and CD8 T cell mediated graft-versus-host disease, again, without global immunosuppression. We see decreased secretion of inflammatory cytokines such as TNF-alpha and interferon gamma. We see an increased expansion of pre-existing Tregs, which are present in the graft at the time of transplantation. We see markedly decreased gut infiltration by T cells. I'll come back to that because this has been a key observation as well in our non-human primate studies. But we see preserved and enhanced in vivo T cell expansion in lymphoid tissues and largely preserved cytotoxicity and graft-versus-tumor activity. And we also see a very unique transcription program induced by Notch in alloreactive T cells that doesn't overlap with other transcription programs that have been studied in T cells downstream of other signaling pathways. So the next step in our studies was to think about translational opportunities based on these genetic observations. And so we wondered if it would be possible to target therapeutically individual Notch ligands and receptors to prevent graft-versus-host disease. And we turned to individual ligands and receptors because in data that, again, I don't have time to show you, we found that administering gamma secretase inhibitors, for example, a very broad pan-Notch inhibitors, was poorly tolerated early after transplant. And so we thought that targeting just the individual ligands and just the individual receptors that mediate graft-versus-host disease could be an alternative approach. And in a lot of work that I'm summarizing here, we zoomed in on just 2 of the 5 mammalian Notch ligands, Dll1 and 4, and just 2 of the 4 Notch receptors, Notch1 and Notch2, as the critical drivers of graft-versus-host disease. The winning approach from a translation perspective ended up targeting the ligands, and you can see the result of a key experiment on that slide when we target either Dll1 alone with monoclonal antibodies, Dll4 alone with monoclonal antibodies, or both Dll1 and Dll4 together, as compared to a genetic intervention of blocking Notch permanently in T cells. And you can see high mortality when wild-type T cells are administered in the presence of isotype control antibodies, some modest protection with Dll1 blockade alone, some higher protection when blocking Dll4 alone. but a much higher degree of protection, approximating the protection that we achieve with genetic methods when we both block both Dll1 and Dll4 ligands early after transplant. And to our surprise, we tried different regimens of anti-Dll1 and Dll4 blockade. And even in this disease, which is mediated by T cells and has a chronic component, we could see very sizable long-term protection with even a very short course of Dll1 and Dll4 blockade around the time of transplant, about 10 days in this particular case. So to study the impact of Notch during this very early window of transplantation, we decided to identify the critical time window in which the Notch signals are absolutely required to trigger graft-versus-host disease pathogenesis. So we modified the administration schedule of the Dll1 and Dll4 blocking antibodies and skipped the first 2 days post-transplant, leaving Notch signaling intact for 48 hours, and then blocked Notch signaling. Or we administered the Notch ligand blocking antibody as a single dose at day 0 only. You can see that when we block all the way through, starting at day 0, we can profoundly decrease the production of inflammatory cytokines by T cells, in this case CD4 T cells in the spleen. At day 6 after transplantation, markedly decreased production of TNF-alpha, interferon gamma, IL-2, and interferon gamma in this particular example, and markedly increased expansion of FOXP3 expressing regulatory T cells, both in relative numbers and in absolute numbers. But when we skip the first 2 days of inhibition and we leave Notch intact for 48 hours after transplant, you can see that we largely lose the protective activity of Dll1 and Dll4 blockade. So persistent production of inflammatory cytokines and no expansion of regulatory T cells. So the first 48-hour window is absolutely critical for the impact of our intervention in acute graft-versus-host disease. And that translates into long-term effects as well, because if we skip again this early window in purple, you can see that the mice have persistent high degree mortality with only a very minimal amount of protection and a high degree of graft-versus-host disease using clinical criteria. And then on the opposite, you can protect the mice with just a single dose of Dll1, Dll4 blocking antibodies at day 0 with equivalent activity as more prolonged courses. So this very early window seems to prime T cells to become pathogenic in graft-versus-host disease. We wanted to understand where the ligands were coming from during these very early steps after transplantation in mice. Now, initial hypothesis was that the ligand would be provided by professional antigen-presenting cells, hematopoietic cells that are still present in the host at this early time point. But to test it formally, we used bone marrow chimeras that are described on this slide. We obtained MX3 positive Dll1, Dll4 conditional knockout mice from our collaborator Freddy Ratke in Switzerland. We induced loss of Dll1 and Dll4 by poly-IC injection. And then we created syngeneic bone marrow chimeras across the CD45 to CD45-1 congenic system to replace all the hematopoietic cells with cells lacking Dll1 and Dll4 or control MX3 positive cells. We waited for full reconstitution to have stable bone marrow chimeras. And then we used these bone marrow chimeras for a second transplant, this time an allogenic transplant using biopsy T cells and biopsy bone marrow as a donor, or a syngeneic transplant as a negative control. And so the prediction was that if the critical source of Notch ligands was indeed hematopoietic, any type of hematopoietic cells, these bone marrow chimeras should be protected from graft-versus-host disease. And to our surprise, we observed no protection whatsoever by deleting Dll1 and Dll4 solely in the host hematopoietic system. This is shown here through this survival graph. The key group is here in red, Dll1, Dll4 knockout chimeras receiving a control, ice-type control antibody, showed very high mortality, very similar to the mortality of control bone marrow chimeras made with wild-type donor bone marrow cells. And in contrast, both groups could still be protected by systemic administration of Dll1 and Dll4 blocking antibodies, indicating that the model remains sensitive to Notch signaling, but the critical source of Notch ligand is not in the radiation-sensitive host hematopoietic cells. So we did a lot of experiments to look for the critical source of the Notch ligands and ended up zooming in on radiation-resistant non-hematopoietic stromal cell subsets in secondary lymphoid organs as the critical source of ligands in this context. And so in lymphoid organs, this is a lymph node here that is schematically represented. You have a variety of different specialized stromal subtypes. You have blood endothelial cells that mediate entry of B and T cells into the lymph nodes. You have lymphatic endothelial cells. You have follicular dendritic cells in germinal centers, marginal reticular cells that line the marginal sinus, and fibroblastic reticular cells, actually a range of fibroblastic reticular cell populations, predominantly in the T zone or at the TB border, a vast number of cells that are being increasingly recognized in immunology to have potent and specific immunological functions. And we were lucky that these three cell types, the follicular dendritic cells, the marginal reticular cells, and the fibroblastic reticular cells, can be specifically lineage traced with a back transgene where Cre recombinase is driven by CCL19 regulatory sequences. This is a very precious reagent in the field that was created by our colleague Burkhard Ludwig in St. Gallen, Switzerland. And when we knocked out Dll1 and Dll4 specifically with the CCL19 Cre transgene, erasing Dll1 and Dll4 uniquely in this population of fibroblastic stromal cells and nowhere else in the body, we could completely protect the mice from graft-versus-host disease with very high degree of survival as indicated here, and only limited clinical activity, in this case indicated by weight change in the red curve, as compared to the group of mice receiving T-cell depleted bone marrow only, with even a better protection than the one we had seen by blocking Dll1 and Dll4 transiently with monoclonal antibodies. So this rare population of stromal cells turned out to be the very unexpected but interesting source of Notch ligands in graft-versus-host disease. So we are now studying this population of cells in great detail in the lab in work that is ongoing, but I'm going to show a few snapshots of the things that we are doing to understand the biology of these cells. If you take collagenase digested lymph nodes and you look at the rare population of cells that are non-hematopoietic, CD45 negative, and negative for the erythroid marker TER119, you can subdivide these populations in 4 different populations based on the expression of the endothelial marker CD31 and the fibroblastic marker protoplanin. The fibroblastic stromal cells in general are characterized as CD31 positive, CD31 negative, protoplanin positive, and a fraction of these cells, roughly 50 to 60% of them, are lineage traced with the CCL19 Cre transgene. They can be lit up by crossing the CCL19 Cre to a ROSA26 EYFP reporter. This must contain, based on our genetic information, the critical source of Notch ligands in graft-versus-host disease. And indeed, a fraction of these cells is labeled by a Dll4 and Cherry reported transgene that was generated by our colleague Yanis Aifantis at NYU and generally shared by him. So these are the candidates, Dll4 positive fibroblastic stromal cells that represent this critical immunological niche in graft-versus-host disease. We can also visualize the location, the physical location of these cells, and we are doing also work now to image the co-localization of alloreactive T cells with these cells in the spleen and in lymph nodes. This is just an example here showing spleen images that report for CCL19 Cre ROSA YFP in green. In the white pulp, you can see B cells and T cells here in blue, and in white, in a white pulp, in a lymphoid follicle in the spleen. You can see now expression of the Dll4 M cherry here on the bottom, Dll1 M cherry here on the top. You can see that the pattern is different. So the populations that express one versus the other ligand actually seem to be different. And when we look for co-localization of the green marker and the red marker here in yellow, we can define the candidate populations of stromal cells that are lineage traced with CCL19 Cre and express our Notch ligands with borders. And you can see without going into the details, that there is in particular a population of cells very close to the marginal sinus at the interface with the red pulp and slightly more internal for Dll4 that are high expressors of Dll4 and are prime candidates as a source of Notch ligand. There are also other population of cells more deeply in the lymphoid follicle. This is all ongoing work. Then interestingly, this is also a regulated phenomenon. So if we look at the expression of the Dll4 protein at the surface of these fibroblastic stromal cells, this is done here by flow cytometry, you can see relatively limited expression on baseline. But at 12 hours after transplantation, right in the middle of this peak of sensitivity to Notch inhibitors, you can see that a population of CD157 high fibroblastic stromal cells, which is enriched in T-zone fibroblastic reticular cells and marginal reticular cells, highly upregulates Notch ligands. This is actually an upregulation that's present only in the allogenic transplant setting and only when we inject alloreactive T cells into these mice. We think it is the product of a crosstalk between the incoming alloreactive T cells that get activated in this host and talk back to the stromal cells, leading to upregulation of Notch ligand expression. So without going into further detail, since this is a snapshot of ongoing work, I want to conclude the first part of this talk by showing you what our revised 2021 working model is. We think that we are dealing with newly identified immunological stromal niches in secondary lymphoid organs that are made by a specialized population of fibroblastic stromal cells expressing Notch ligands that closely interact with T cells very early after transplantation. We think this happens in parallel to their exposure to alloantigens by other cell types, and that this interaction has a profound impact on the eventual outcome of the immune response that is triggered by bone marrow transplantation. So I want to spend the next 5 minutes or so discussing some of our more recent translational work in non-human primates. This has been a wonderful collaboration with Leslie Keane, who used to be with you at Emory before she moved to Seattle, and now as the chief of pediatric bone marrow transplantation at Boston Children's. And the key question that we wanted to address with Leslie is, what is the potential of Notch pathway inhibition to prevent graft-versus-host disease in patients, and specifically to address whether our observations in mice would translate to higher organisms. And the model that Leslie developed has a lot of advantages to address this question. It's a T-cell replete haploidentical transplantation model in non-human primates using the rhesus macaque as a whole. Is very similar to human transplantation, but it has the unique potential to assess single agent activity of new candidate therapeutics, something that could not be done, of course, in human patients as an initial clinical trial. This is how the model is run by Leslie as her team. So this is using a GCSF mobilized leukapheresis graft. The conditioning regimen is total body irradiation, so lethal irradiation. It's a high T-cell rich leukapheresis graft. And then the animals are being supported by intensive supportive care that include daily labs, daily antibiotics, antifungals, etc., with or without any additional intervention. And in this case, the intervention that we study is blockade of the dominant Dll4 ligand, which we found to be the most important driver of GVH in mice. And this is work, again, in collaboration with Leslie Keane and Viktor Tkatchev at Boston Children's, with a very heavy support from a team at Regeneron that has produced this anti-Dll4 antibody. And so these are the overall results of the experiment in comparison to other single agent interventions that Leslie has studied in the past in this model. So in the absence of intervention with no prophylaxis, this is a rapidly lethal model of hyperacute graft-versus-host disease with a median survival time of only about 8 days before the animals meet euthanasia criteria. A very rapidly lethal model in this case. And you can see that some of the interventions that Leslie has studied that are actually making their way now into clinical trials have had some degree of activity, a little bit of activity from CTLA4 Ig, a little bit of activity from sirolimus, a little bit more from the OX40 ligand blockade. And out of all these interventions, the blockade of Dll1 alone with a single injection of the antibody at day 0 ended up having the largest increase in median survival with animals that even survive all the way to day 40. So this is not a model that is meant to treat the graft-versus-host disease that arises in these animals, but that's clearly consistent with a high degree of single agent activity of Dll1 blockade in this model, basically a conservation of the findings all the way from mice to non-human primates. And one of the striking observations that Leslie and her team made is that unlike other interventions that they had studied in the past, the animals receiving anti-Dll1 blockade appeared to have a very high degree of long-lasting protection from gastrointestinal graft-versus-host disease, essentially a stone-cold clinical score for gastrointestinal symptoms, absolutely no diarrhea at all the way through the entire procedure, very much unlike any other interventions that they have studied in the past. And in pathological scoring, blinded pathologists could not differentiate the gut of Dll1 treated non-human primate recipients from that of control animals that had not been transplanted. There was less protection in other organs, so still skin graft-versus-host disease, liver graft-versus-host disease at the time of euthanasia, but the protection in the GI tract was very impressive. We are looking at that in more detail right now using both data sets in non-human primates and experiments in mice where all of this is actually conserved. And one of the striking observations is a decreased expression of the integrin dimer alpha-4 beta-7, which is very important for the homing of T cells to MADCAM expressing vessels at the base of intestinal crypts, as shown by many investigators, including Alan Hanash, for example, at Memorial Sloan Kettering. And you can see that this is found in many different systems. And we've seen in the data I'm not showing here that this is also conserved all the way from mice. And this translates into decreased accumulation of T cells at the base of intestinal crypts, as shown here on these immunofluorescence studies in animals with GVH, large accumulations of T cell subsets at the base of intestinal crypts. And this is controlled in Dll1 treated, Dll1 blocked animals to reconstitute a pattern that looks much more like healthy controls. So in summary for this part, I've shown you that specialized fibroblastic stromal cells in secondary lymphoid organs are the essential source of Notch ligands in vivo at the onset of graft-versus-host disease. This is building on the emerging concept that these stromal cells form very specialized and active immunological niches in secondary lymphoid organs with profound impacts on immune responses, as shown here in graft-versus-host disease. I've shown you that short-term inhibition of these Notch ligands induces long-term protection from graft-versus-host disease. And importantly, that transient blockade is sufficient to provide high-level protection from gastrointestinal GVH in non-human primates. This is conserved during evolution, and it has translational potential. So during the last 20 minutes or so of the talk, or 15 or 20 minutes, I want to share some new insights from the lab and from other investigators that suggest that these stromal cells I just told you about in the context of graft-versus-host disease also have a major role to play in other contexts that are relevant to human disease and human cancer, specifically an oncogenic role in human lymphomas and a role that is active in stroma-dependent processes in normal and malignant B cells, basically where the B cell lymphomas that rely on Notch signaling borrow from the playbook of mature B cell homeostasis. And so for the oncogenic function of Notch in lymphomas, I want to briefly review the genetic information that's available in the literature. The first oncogenic role of Notch in human cancer was documented back in 1991 in T-cell acute lymphoblastic leukemia, based out of rare translocations, and then more recently in 2004, out of the identification of point mutations in the Notch1 receptor gene in up to 60 or 70% of cases in T-cell acute lymphoblastic leukemia. In B-cell lymphomas, the discovery is more recent, but activating mutations of Notch1 and Notch2 are found in about 5 to 25% of chronic lymphocytic leukemia / small lymphocytic lymphomas, marginal zone lymphomas, some mantle cell lymphomas, and rarely other types of lymphomas, but predominantly in CLL and marginal zone lymphoma. This is data from a large number of groups that are listed here on the bottom. There are unique features to the mutations that are found in B-cell lymphomas as opposed to those found in T-cell acute lymphoblastic leukemia. So many of the mutations in T-cell acute lymphoblastic leukemia destabilize the extracellular region of the Notch receptor and lead to ligand-dependent activation of the pathway. In contrast, all of the mutations, virtually with no exception in B-cell lymphomas, are truncation mutations that remove a C-terminal pest sequence that is important for degradation of the active Notch molecule. So the predicted end result of these truncations is an increased half-life of the active intracellular Notch. This is also something that is predicted to be ligand dependent. There would not be any oncogenic activity if there was not a proximal ligand receptor interaction that triggers activation of the Notch receptor in the first place. There's additional data from John Astor's group in Boston that suggests that this activation uniquely happens inside secondary lymphoid organs and inside lymphomas. So this is a very interesting paper in PLOS One in 2013 from John, where he measures active Notch using immunohistochemistry. The brown staining here identifies cleaved active Notch molecules. And this is a case of CLL, where the disease extends outside of the lymph node capsule here in the fat that surrounds the lymph nodes. And you can see very high Notch activity in the lymph node with limited activity to no activity as soon as the same lymphoma cells reach the lymph node capsule. You can see the same phenomenon in this case in angioimmunoblastic T-cell lymphoma, a T-cell lymphoma that has known activating Notch mutations, where activity is very high within the lymph node and very limited outside of the lymph nodes. That's suggestive that these cells are seeing Notch ligands inside the secondary lymphoid organ. And then more importantly, there are some recent data from Ricardo de la Favra in PNAS 2017 and a recent paper by John Astor in Blood Advances that just got published 10 days ago in marginal zone lymphoma, indicating that Notch can be very commonly activated by non-mutational mechanisms in chronic lymphocytic leukemia and marginal zone lymphoma. This is indicated here in CLL, where you can see that the large fractions of CLL in red that have evidence of active Notch are actually non-mutated wild type for Notch. And if you look at these data, you can see that approximately 2/3 or so of CLL patients have evidence of active Notch activation, of Notch mutation, non-mutational activation. In other words, non-mutational Notch activation may be the hidden part of the iceberg, and a very significant fraction of patients with CLL actually have Notch engaged as an oncogenic pathway, much more than you would predict just from the frequency or the prevalence of mutated patients. Now, our interest into the field got also captured by this very interesting paper from Brad Bernstein, John Astor, and Warren Pear's group in 2017 that identified a transcriptional program controlled by Notch in small B-cell lymphomas using the very few cell lines that are available to study this pathway in human cell lines. And this includes a control of MYC, a control of cytokine responses, a control of BCR signaling, and other pathways that are known to be important in B-cell lymphomas and could represent important therapeutic targets. Now, our interest was particularly triggered when we realized that this transcriptional signature that this group had identified in human B-cell lymphomas was highly conserved in Notch-dependent B-cell subsets, normal Notch-dependent B-cell subsets that we were studying in mouse lymphoid organs. So there's a subset of mouse B-cells called marginal zone B-cells that's highly dependent on Notch ligands coming from the stroma of lymphoid organs. And if we look at the transcriptional program controlled by Notch in human B-cell lymphomas, about 70% of that program happens to be regulated by Notch as well in mouse marrow B cells. So very highly conserved Notch signature in the B-cell lineage, all the way from normal B cells to malignant B cells. So I want to show you some ongoing data from the lab that characterize the role of Notch signaling in these B-cell subsets as they inform us about conserved features of the program controlled by the pathway that are hijacked in B-cell lymphomas. So in mouse, you have two main populations of mature B cells. And in the spleen, they can be divided into a population of follicular B cells, which are deep in the follicle and are the most abundant population of cells. It's a circulating population of cells that participates in particular in germinal center reactions. And then there's another population of cells that's very specifically localized at the interface with the red pulp here in green, outside of the marginal sinus and the marginal zone layer, where marginal zone macrophages are being located here in red. So these would be the marginal zone B cells. This is a very interesting population of cells that's innate-like, that can make plasma cells very rapidly, that's pre-activated and specifically segregate into the marginal zone of the spleen as a non-circulating population of cells. The traditional view of Notch's role in that population is based on papers from two groups in the early 2000s that documented a critical role for Notch2 in the generation of marginal B cells, with the concept that Notch can promote the generation of marginal B cells and block the generation of follicular B cells, essentially as a binary cell-fate decision. We think instead that beyond development, Notch signaling regulates the maintenance and the homeostasis of the mature B cell pool at steady state and during lymphopenia. And these critical physiological functions are very precisely hijacked during the process of Notch-driven lymphomagenesis. So if we block Notch signaling in vivo acutely and look at the impact of Notch signal on marginal B cells, this is indicated here schematically. We are using blocking antibodies against either Notch receptors or Dll ligands. The key ligand receptors in this case are Dll1 and Notch2. And we can use a time course to measure the impact of Notch signaling on different B cell populations. We observe a very rapid disappearance of the marginal zone B cells, as indicated by this flow cytometry graph after Notch2 blockade in vivo. You can see that the numbers of marginal zone B cells, as indicated by a CD21 high, CD23 low phenotype, normally about 8% or so of the B cell pool, of the mature B cell pool in the spleen. These numbers rapidly drop already detectably at 12 hours after Notch inhibition, more significantly at 24 hours, down to essentially zero about 72 hours later. This is quantified here. And at the same time as these numbers drop in the spleen, we see release of cells that look like marginal zone B cells, which are normally very low in the blood because this is a non-circulating population, but they peak at around 48 hours after Notch inhibition. And moreover, when we look specifically in the spleen, we can see that marginal B cells here indicated in white through their high expression of CD1D are normally tightly associated with the marginal sinus. But when we block Notch signaling, in this case, 24 hours and 48 hours later, you can see that even before they have physically disappeared, you can still see phenotypic marginal zone B cells at 24 hours and 48 hours. They have already relocated away from their tight association with the marginal zone, in particular relocated into areas of the red pulp, indicating that Notch is acutely required for the retention of marginal zone B cells in the spleen marginal zone. We find a very interesting transcriptional program regulated by Notch using RNA-seq at 12, 24, and 48 hours after Notch inhibition. This is comparing marginal zone B cells on the top and follicular B cells on the bottom. You can see a large number of transcripts regulated by Notch initially. A lot of known Notch targets already being downregulated at 12 hours that include Deltex, HES1 and HES5, and then MYC, which is this very highly conserved target in human lymphomas, and eventually the number of genes that get dysregulated, the increases in marrow zone B cells as they are being lost, while it remains much lower in follicular B cells, consistent with the high sensitivity of marrow zone B cells to Notch signaling. These genes can be segregated into different functional pathways. So we see a very clear mixed signature in marrow zone B cells, which we think is functionally important for some of their function as pre-activated cells that can rapidly differentiate into plasma cells. We see a really interesting chemokine receptor and integrin signature, and we see also survival and apoptotic genes being regulated by Notch. Another really interesting setting where we think that the effects of Notch are even magnified to a larger extent, and again, where Notch fulfills function that are then hijacked during lymphomagenesis, is in the context of lymphopenia. We can model that by transferring highly purified follicular B cells into either lymphoid replete C57 black 6 hosts or lymphopenic RAG2 knockout recipients that lack any T or B cells. We can track these recovered T cells at different time points after transplantation, and we can use flow cytometry to characterize their phenotype and differentiate a phenotype that looked like follicular B cells. This would be here in black, with high expression of CD23 and IgD, low expression of 21 and CD1D or IgM, or a phenotype that would look like marrow zone B cells. There would be low expression of CD23 surface IgD and high expression of CD21, CD1D, and IgM. And when we do this transplant and look at day 2, we can recover the cells at day 2. They still look largely like the initial follicular B cells that we have injected both in the lymphoid replete and the lymphopenic recipients, except that CD21 starts to be upregulated, and CD21 happens to be a direct transcriptional target of Notch signaling. You can see this upregulation here in red in the follicular B cells that have been transferred into the lymphopenic RAG knockout mice. When we follow the fate of these cells at day 4, you can see that this upregulation gets magnified. Now CD1D gets upregulated, surface IgM gets upregulated, CD23 starts to be downregulated. The cells start to shift into a phenotypic criteria of marrow zone B cells. And then they go into a burst of proliferation, and they further change their signature, again, specifically in the RAG knockout recipients, not in the lymphoid replete recipient, and start acquiring a cell surface phenotype in red. It's very reminiscent of what marrow zone B cells look like. So we think that this is a transdifferentiation of these follicular B cells into marrow zone B cells. Or marrow zone-like B cells that's followed by a burst of proliferation. We can see that these cells acquire cell surface integrins and cell surface chemokine receptors that are also characteristic of marrow zone B cells. This is shown here in RAG2 knockout recipients. The cells upregulate LFA1, which is an integrin that marrow zone B cells use to be retained in the marginal zone. They upregulate S1P receptor 1 and CXCR5, which are also typically expressed by marrow zone B cells. And they downregulate CXCR4, which is expressed by follicular B cells and suppressed in marrow zone B cells. And they also locate into the area that resembles the marginal zone area in these mice. You can see here in the RAG2 knockout recipient on the right, the marginal zone layer is identified by this cluster of CD167 positive macrophages. You can see the transferred cell here in blue, locating, clustering around this layer here. This is not present at all in the lymphoid replete recipients. And eventually the cells proliferate in that area and remain tightly associated with this marginal zone area at day 8 after transplantation. So it turns out that this process is entirely Notch dependent, as you may have surmised from my introduction. So if we block Notch2 with monoclonal antibodies, the same monoclonal antibodies from Genentech that I've shown you before, we can do that by comparing isotype control treated hosts and anti-Notch2 treated hosts. You can see here on the bottom that we see this transdifferentiation of the follicular B cells into marrow zone B cells when the cells are transferred in lymphopenic recipients in the presence of Notch signaling, in the absence of antibodies against Notch2. When we block Notch2, this transdifferentiation is completely abrogated. And furthermore, when we now when we do the experiment genetically, we know that to look at the source of Notch ligand, we know from our colleague Freddy Radke that when we knock out Dll1 specifically in this population of stromal cells in the spleen, we completely lose steady state marrow zone B cells. And it turns out that when we knock out Dll1 now on a RAG2 knockout background and then transfer follicular B cells, the vast majority of this transdifferentiation is also abrogated simply by the loss of Dll1 in the stromal compartment. So we think this is consistent with the stroma, with the injected follicular B cells sensing the degree of lymphopenia through their encounter with stroma-derived Dll1 in the host in this particular context. And we think that this is consistent with a new working model that revises the classical model of the Notch function in binary cell fate decision into a more complex function of Notch in these cells, in which Notch controls not only their development, but also their maintenance, their positioning, their function, and then regulates also the size of and the composition of the peripheral B cell pool through stroma-driven transdifferentiation of follicular B cells into a margin zone B cell pool under the control of Notch. And we think that many of the features of this program, again, are prime targets for being hijacked during transformation in stroma-driven, Notch-dependent B cell lymphoproliferative disorders. So in summary for this last part, I've shown you that stromal Notch ligands control a highly conserved transcription program in B cells, and that this signaling is enhanced in B cells when they are sensing a lymphopenic environment. I've shown you that, contrary to what has been commonly assumed, follicular B cells are not locked in their cell fate, but are endowed with a plastic transdifferentiation potential, in this case, with a Notch signal functioning as a checkpoint for this transdifferentiation. And I've shown you data that Notch-driven B cell lymphomas borrow extensively from the physiological functions of Notch signaling. As we think this is creating new experimental approaches where studying the roles of Notch, the physiological roles of Notch in B cells, ends up providing information about its role in transformation in diseases that are actually quite difficult to study experimentally. So this is to the end of my, coming to the end of my talk. I'd like to thank members of my lab, shown here in a pre-pandemic photograph, were listed on the left. A lot of work here that I mentioned by many members of the lab, but in particular Leolyn, Daniela, Sam, Ashley, and several previous members of the lab. We've had several wonderful collaborators at Penn, in particular for this B cell project, which emerged after I moved back from the University of Michigan to Penn in 2018. We have wonderful collaborators at Boston Children's, Victor Tkachev and Leslie Keane, our collaborators at Regeneron in Switzerland, at Genentech, Bruce Blazer at the University of Minnesota, and other collaborators who I largely mentioned along the way. And then finally, funding from the National Institutes of Health and the Leukemia and Lymphoma Society. Thank you for your attention.
Ivan, thank you very much. That was really a stupendous overview of Notch, both in immunological settings, in B cell development, and in oncology. We have a couple of questions in the chat box. First from Andreas Chang. How does delta-like 1 or 4 blockade affect the anti-tumor effect of allogeneic transplant?
Yes, we've tested that and published that in our JCI 2013 paper. It was very similar to what we have observed with genetic blockade of Notch signaling in T cells, in the sense that we saw largely preserved graft-versus-tumor activity. If we pushed the dose of leukemia lymphoma cells to extremely high levels, we could see some leukemias, break the barrier of the graft-versus leukemia activity. But the degree of preservation was very high with Dll1 and Dll4 blockade, just like it was with genetic blockade in T cells.
So that brings kind of the broader question of why T cells that are alloreactive and might cause graft-versus-host disease are stopped and their activation pattern is interrupted, but T cells that presumably mediate anti-infectious protection or anti-tumor activity, their activity is not checked. What's the difference between the fates of those different types of T cells?
Yeah, this is something that we are actively studying and we have some answers for, but we are still doing work. And I think one of the main things is the trafficking effect of Notch inhibition, the trafficking impact of Notch inhibition. So one of the dominant mechanisms for graft-versus-host disease protection in mice, and now we are seeing that also in the non-human primates, appears to be to either prevent their trafficking into the gut or prevent their homing and retention in the gut, or to tilt the ratio of regulatory T cells to conventional T cells in the gut. But in any case, really protect the key early target organ of graft-versus-host disease from immune attack. While if you look at secondary lymphoid organs, the expansion of alloreactive T cells is actually completely preserved or even enhanced in the absence of Notch signaling. That's very different from what you would see with any global immunosuppression. So I think that's the first reason, that you actually do create a pool of alloreactive T cells in lymphoid organs where they can exert cytotoxic activity. The second reason, if you look at their expression of cytotoxic molecules, we do see a mild reduction in gene expression of things like granzyme and perforin, for example, but it's only very subtle as compared to the profound reduction in pathogenic cytokines. For example, TNF-alpha is markedly decreased, interferon gamma is markedly decreased by Notch inhibition. The cytotoxic pathways are better preserved. So we think that altogether, and then the expansion of Tregs is probably another reason why we see protection from graft-versus-host disease. So we think this is probably a combination of reasons why we affect the graft-versus-host disease pathogenesis much more profoundly than we do affect the ability of these cells to kill tumor targets.
So it's not deletional tolerance, it's an absence of retargeting the T cells to the GVHD organ.
Yes, exactly. We don't think that we are reaching actually true tolerance by blocking Notch signaling. We think we are modulating their effector activity and we are modulating their trafficking.
And if you retransfer those T cells in a Notch deficient animal to a third party, do they retain their alloreactivity and cause GVHD?
Yeah, that's an interesting experiment. We haven't actually done that, Ned. I think that's something that we could try. I suspect they would, but it's a little bit of a complicated experiment because you don't have a lot of naive T cells left, right? Which are the dominant drivers of graft-versus-host disease in mouse models. A lot of them are going to be effector or effector memory. So it's not completely trivial to do that. I suspect that just by itself, Notch may not be sufficient to trigger tolerance. Now, there are some data in the context of solid organ transplantation that are being generated, and particularly a really interesting paper from Leonardo Riella's group at Harvard in Circulation, I think last year, that showed that when you combine Notch blockade with interference with the B7-CD28 pathway, you can actually something that looks like actual tolerance in solid organ transplantation. But it doesn't look like Notch inhibition by itself is sufficient to achieve that goal. So I think the jury is still out, but I don't think we are seeing true tolerance in our transplanted animals.
CK asks whether you've looked at graft-versus-tumor and the CCL9 positive cell-specific Notch ligand knockout mice?
We haven't done this in these mice. We have done it in the antibody-treated mice. We suspect it would be similar, but we haven't actually studied graft-versus leukemia in the CCL19-3, Dll1, Dll4. We've done it with the antibodies.
And have you looked at immune responses to other things in your model systems like vaccines or CMV infection?
We've done a little bit of that work. It's complicated to do in the post-transplant setting. So we had a collaboration back at the University of Michigan to look at that. It's complicated by the fact that the mice that get graft-versus-host disease end up being very, very sensitive to viral challenges. They are just sick in the first place, and then you get the viral infection and they die. And so it's really hard to see if you can, to assess the contribution of T cell function to that in general. But we are doing experiments outside of transplant to look at the effects of Notch in antiviral responses. This is something we're doing in collaboration with Nathalie Labrec at the University of Montreal. And so we are studying LCMV infection. We are studying also listeria models, which are driven by, controlled by CD8-dependent T cell responses. And we can see selected effects of Notch in that context. So it looks like Notch is important for the differentiation of short-lived effector cells, very high T-bet interferon gamma expressing CD8 T cells that are present during the very acute infection. But we don't see an effect on memory precursors or on other cell types. And we don't see an effect on the overall magnitude of the size of the tetramer positive antigen-specific T cells in that context. So it looks like more selected effects on aspects of the immune response and not a complete loss of the antiviral activity.
And we're running a little bit over, but I'll indulge myself with this opportunity to ask another couple of questions. If you were to look at Notch in the hierarchy of all the molecules that have been studied in GVHD, co-stimulatory blockade, CTLA-4 Ig, calcineurin inhibitors, where would you place it? I mean, clearly the clinical practice has evolved to use calcineurin inhibitors, methotrexate, because of the availability of those drugs. They still remain the mainstay of prophylactic therapy. Other drugs are kind of moving up, like abatacept or belatacept. But putting aside what practice is, just from the principles of immunology, where is Notch in that hierarchy?
Well, that's a great question. We've really thought a lot about it recently, in particular with Leslie, and then thought about where we should go in terms of further translational development in humans on that basis. So the emerging data we have with Leslie using transcriptomics in non-human primates show that Notch acts differently from all the other interventions that have been studied so far in her model. So the transcriptomic signature in T cells from our Dll4 block monkeys looks very different from the transcriptomic signatures of monkeys receiving B7 CD28 blockade, for example, or OX40 ligand blockade. And we need to generate data with calcineurin inhibitor treated monkeys, but we would also expect this to be different. So I don't think that this is going to be a matter of potency as compared to the other agents. It's going to be acting differently. And so how we integrate that with existing treatment is going to be the interesting questions. And we think that the most interesting situation would be a situation where gastrointestinal graft-versus-host disease is the dominant problem. And because that's what Notch is really, really good at preventing. And this is something that could be prevented potentially even without other prophylactic immunosuppressive drugs. So we are working on some concepts actually along those lines to see if the Notch inhibitors could be deployed in that particular concept. We think it would be more interesting to do it this way rather than have them compete against other immunosuppressive drugs. We think it may require to deploy them in a unique niche, actually.
So you're thinking about local administration then to the gut?
No, we're working with a team at the Dana-Farber on a model of transplantation, which would be calcineurin inhibitor free, where the dominant remaining problem is graft-versus-host disease. So we think that the Notch inhibitors in that context may turn out to be an interesting strategy. That's the idea. It's still work in progress. It's still in the planning phase, but that's what we're thinking at this point.
Well, thank you, Ivan, for your time, your science, and your clarity in this great exegesis of Notch. I think it remains for me at kind of the pinnacle of immunological molecules with its so many diverse functions and regulatory pathways. And I think this is also such a nice example of translational science that we should all look to emulate. So thank you, and thank you for the participants and in their questions, and I hope to see you soon in the flesh, maybe again in China.
Thank you so much, Ned. Thank you for the invitation and for the great questions. I really
all right.
Thank you, everybody.
Bye-bye.